Hi, my name is Sarah Pilling, and my project is about the abundance and distribution of brown stingrays off the coast of Santa Catalina Island. So, a little bit of background about brown stingrays is that they comprise the highest biomass of any nearshore fish off the coast of California, making them very ecologically important. They also group in formations called aggregations, which can be defined as two or more individuals at the same place and time, usually due to a common driver. And they are also known as ecosystem engineers, which means they physically affect the environment around them, also contributing to their ecological significance. So brown stingrays are benthic in fauna feeders. So what they do is they feed on prey on the seafloor through suction feeding, which leaves small depressions in the seafloor called feeding pits. And this plays a role in bioturbation, which is when animals disturb sediment through burrowing, digging, feeding, or living in it. So other examples can include how earthworms will create tunnels in soil, or how crabs will dig or burrow in sand. And with the case of, and this process contributes to restratifying sediments, oxygen penetration, and nutrient cycling. So round rays' role in bioturbation is important because the feeding pits that they create are deep enough to disturb the rhizosphere of seagrass, which helps the seagrass, which aids in seedling recruitment for seagrass meadows. And this is important because seagrasses not only provide nursery habitats, but they also improve water quality, and they constitute a globally important carbon sink. And on the Pacific coast of the United States, both state and federal governments consider seagrass a critical habitat with high priorities for conservation. So the goal of this study was to see how habitat type affects the distribution of brown stingrays off the coast of Catalina Island. This is because where the brown stingrays are and what environmental pressures influence that can provide very valuable insight for managers when thinking about the conservation of this species and the other species that it interacts with. So how did I actually do this study? So what I did was I had what's called a transect tape, which is that thing in the corner, and it's basically like a giant ruler with every meter is marked. So I would swim one of those for 30 meters along the seafloor, and at each meter I would record what substrate was below. So whether it was seagrass, mud, sand, or even cobble or algae. And then if 75% or more of the points were seagrass, mud, or sand, the transect would be qualified as a transect that was ran in that habitat. And then after doing this, I would swim back, and for each meter from the left and right of the transect, I would record the number of rays I saw, and if they were in an aggregation, how big the aggregation was. So this gave me a measurement for density by taking the transect area totaled 60 meters squared, because 30 by 2 meters. And so I would have the total number of rays seen within that 60 meters squared, which gave me an estimate for density. And then I would also have the total number of aggregations I saw in that area, and the total number of single rays, and the size of each aggregation that I saw. Oh, and the deep transects, so the seagrass ones, were done on scuba, and the shallower ones were done on snorkel for sand and mud. Okay, so here's where this study took place. You can see there is the Long Beach port, and then off the coast is Santa Catalina Island, about 26 miles from shore. And then this tiny part of Santa Catalina Island is where there were 3 coves, and all of the transects took place within those 3 coves. Okay, so as I mentioned, we measured for density, which is the rays per meter squared. So this is an example of if you saw 3 rays within 9 square meters, you would get an estimate of 0.3 rays per meter squared. Aggregation count, so this 3 rays, if they were within less than 1 meter apart from each other, that would be considered 1 aggregation of 3. So the number of aggregations, the size of the aggregation, and then number of single stingrays that were not within 1 meter of any other rays. Okay, so for stingray density, we have ray density on the y-axis and depth on the x-axis, and then each point on the scatter plot represents 1 transect, and the substrate that it was done in, the mud transects are represented by circles, the sand are represented by triangles, and the seagrass are represented by squares. And then the temperature is represented, the warmer temperatures are represented by lighter colors, and the colder temperatures are represented by darker colors. So as you can see for density, there was no difference across any of the variables. Temperature, depth, mud, sand, and seagrass didn't have any effect on stingray density. We saw a similar pattern for aggregation count. The number of aggregations on each transect was not affected by temperature, depth, or substrate. For aggregation size, we did see something interesting, which was that sand had significantly smaller aggregations compared to mud and seagrass. We still saw that temperature and depth did not affect aggregation size. And something else that was interesting, although not statistically significant, you can see that the 3 transects with the highest average aggregation size were all done in seagrass, which, and then we saw a similar pattern for single ray count, which was that transects done in sand had significantly significantly more counts of single rays. Okay, so what does this mean? It means that, firstly, sand is associated with smaller aggregations and higher numbers of single rays. And density and aggregation count showed no influence due to substrate. Temperature and depth also showed no effect on round stingrays. It was interesting that the 3 transects with the highest average aggregation size were in seagrass, because that could mean that with a higher data, with more data points, that could have a significant effect. Although, because I did 5 transects per substrate, that it's possible that that just wasn't enough to show that trend. But we would really need more data to know whether those are just outliers, or whether those would have shown significant effects with more data points. And I also think, and I personally think that it would show that effect, because when I saw the rays in the patches of seagrass, there were many of them, and they were very close together, and they were overlapping, which is something that we didn't collect data on, but something I observed, which I didn't observe in sand or mud. So if I were to run this experiment again, I would definitely measure how close the rays are together, and I would also measure for ecotone effects, which is, for example, if there is sand right at the edge of a reef or a seagrass or something, that is where I observed a lot of most of the rays in sand and mud. So if I had a future study, I would record for those effects. Any questions? You can stop the video.